Hey everyone, welcome to chapter 10. So in this chapter, I'm going to be doing or modifying it a little bit, and I'm going to be cutting some of the material out just because I'm going to focus more on what we do in our lab, and the other information is important, but I have to at some point cut some material because otherwise I wouldn't get through all the chapters. So let's look at history first. So we're always trying to classify organisms, which we talked about actually in chapter 9 a little bit, classifying. So in the 1870s, Ferdinand Kohn, which you heard about when we talked about endospores, actually wrote many papers on classification of bacteria based on shape. And so he started classifying organisms based on if they were rod-shaped, if they were spherical, if they were spiral-shaped. This was decent, but it didn't really put organisms into groups that were effective groups. So then in 1908, Orla Jensen, and his name was Sigurd Orla Jensen. He started classifying organisms based on some of their physiological properties. So this was more beneficial than Kohn's idea, though Kohn's idea was still good. Using physiological characteristics and grouping them was beneficial. The problem was they might be grouped based on one characteristic, but they might differ in other physiological properties. So then in the 1930s, two different microbiologists, Albert Klubier and Van Niel, C.B. Van Niel, they started looking at evolutionary relationships. The problem with this was they were interchanging the resemblance and the relatedness. So two microbes might look alike or act alike, but they might not be genetically related. So in the 1970s, Roger Stanier decided that maybe we could look at relationship based on physical characteristics like proteins that they produce, as well as cell wall structures or nucleotide sequencing, which just had started. So we started looking at nucleotides and sequencing DNA. So this was brand new. So then in the late 70s, Woese recognized that within the prokaryotic grouping, the prokaryotic large group, there were two different groups based on ribosomal RNA. And so this led to the 3-domain system that we have today. So the term taxonomy is a science that studies organisms and arranges them into taxonomic groups or different groups based on characteristics that they have in common. We use taxonomy to identify relatedness between pathogenic strains of different species, or to help us identify spoilage microbes. We also use taxonomy to help us identify beneficial organisms, maybe oil-reducing organisms that can clean up oil spills. And there's a lot of different tests we use when we are trying to identify taxonomy. So some of these tests require microscopy. We also use biochemical tests and nucleic acid analysis, as well as serology. And if I can go to the next slide, we'll talk about phylogeny. If I can get it to work. Ta-ta, hello. There we go. All right, so when we talk about phylogeny, we're looking at evolutionary relatedness. So it's important to have organisms that are evolutionarily related be close together because they're similar genetically. So when we talk about a species, typically when you learn about species in a general biology class, you learn that it's a group of organisms that are related enough to be able to sexually reproduce and produce viable offspring. Since prokaryotic organisms don't sexually reproduce, this is not a beneficial definition. So when we talk about species in prokaryotes, we're looking at a group of related strains. So they have to be closely related organisms, similar strains, okay? And a strain is a pure culture isolate within a group, okay, within a species. This helps us to categorize those different organisms. A lot of times, since it's so hard to isolate organisms, we don't group them into the genus or species. We talk instead about organisms based on these informal groupings like lactic acid bacteria or endospore forming bacteria or sulfur reducing bacteria. So we use Berge's manual to help us to identify different groupings of organisms. So Berge's manual was produced in, I think it was around the 1930s or 40s. And since then, there have been multiple editions, new editions of the manual, but it's still being used today to help us identify different species. It not only helps us to identify different species of bacteria, but it also can be used in helping us to produce or to find new organisms and put them into the proper taxonomic groupings. So here are different methods we use to identify microbes. I'll be talking about some of these methods. Some of them I may not talk about, okay? So phenotypic characteristics. So what are their physical characteristics? What do we see? Things like their size, their shape are phenotypic characteristics, okay? Whereas, and so microscopy helps us to see their physical shapes and size. And then we can look at cultural characteristics. So what does their colony tell us? What type of biochemical metabolites are produced? So what are the waste products produced in a biochemical test? What proteins or polysaccharides are identifying these organisms? That's serological testing. We can do protein profiles. We can detect using nucleotide sequencing. So I talked about Sanger technique in chapter 9. And then we can also look at ribosomal RNA. I'm going to stop here because this has been 9 minutes, and we'll get into microscopic structures in our next video. Bye.